So our second young investigator is Susan Bailey, and she'll be speaking to us about experimental and statistical approaches to understanding the processes driving parallel evolution. Susan?
Thanks. Okay. So I'd like to start out by saying thank you to Mark and the Young Investigators Award Committee for the chance to speak with you today. I'd like to talk to you about one of the themes of my research, and that is trying to look at and understand the processes driving parallel evolution. And I've done this using sort of two main approaches, experimental evolution, and now more in my postdoc, I've been focusing on theoretical and statistical approaches to look at experimental evolution sequence data. So as I said, I'm interested in understanding parallel or convergent evolution, and we're all familiar with this idea that in independently evolving lineages or populations, we often see, or we do see the evolution of similar phenotypes. And here we have the obvious example of birds arising across diverse animal lineages multiple times. And as we gain more and more information about the sequences of these organisms, we're starting to learn that a lot of times there's actually molecular parallelisms happening that underlie these phenotypic similarities. And so this is just a neat example of reduced sensitivity to a toxin that is produced by milkweed plants and other plants. And across 4 orders of insects, we've seen the repeated evolution of reduced sensitivity to these toxins. And this sensitivity has been associated with a handful of amino acid changes in the same locations across these orders. And I've seen a number of talks in this conference where we've had examples of these molecular parallelisms coming up over a wide range of different systems. And so I'm interested in trying to characterize what drives parallel evolution in some systems and not in others. And I've done this, or have looked into this using experimental evolution. So this is a great way to try and get at these kinds of questions because we set up a whole bunch of replicate populations. We evolve them for some period of time, and then we can characterize the changes and see whether they're similar or different across populations. So for the experiment I'm going to talk to you a little bit about today, I started with Pseudomonas fluorescens, which is just a gram-negative bacteria that we use in the lab. And what I did was I evolved this bug for about 1,000 generations in 5 different selection environments. And those environments varied in terms of the sugar resources that were available. So there were different types of sugar resources, different numbers of sugar resources, and the arrangement of those sugar resources was variable as well. At the end of the experiment, I characterized fitness and phenotype in terms of the ability of the different populations to use those resources. And I was also able to get whole genome sequences for those populations to compare similarities or differences in terms of the mutations that arose. So just to give you an idea of what happened, just to remind you to start off that the degree of parallel evolution, so the amount of similarity between populations, is going to depend on where you look. This is kind of an obvious point, but I just wanted to show this plot to say that if I had, for the populations that I had evolving all in the same environment, we saw the same increase in fitness. We saw similar changes in terms of their phenotype, in terms of the way that they used resources, their ability to use the sugar resources that were available. But then when you start to get further down into the details, the way that those adaptations happened were a bit variable between environments. And when you start to look at the gene level, there's some overlap between populations, but it's far reduced. So there's about 30% similarity between populations on average. And as I said, I had not just one single selection environment. I had 5 different selection environments. So I'm going to show you some results now focusing at the gene level and look at the variability across those five different selection environments. So I guess the take-home message is that there is variation across selection environment. So the environment that these bugs are evolving in plays an important role in how much parallel evolution you see. So for example, in one environment, you see a high level of parallel evolution. In another, you see little to no parallel evolution. And the question is, of course, what's driving this variation? And so there have been some theoretical models that have tried to come up with factors that might be important in playing a role in driving this variation that we see. In particular, a model by Shevin and colleagues from 2010 suggested starting distance from optimum level of pleiotropy. I'm not going to go into too much of the details, but we tried to look at some of those suggested factors. And for the first one, didn't seem to be any pattern. For the second one, it's really difficult to get a good idea of how much pleiotropy is going on in each environment. So we weren't really able to answer that question. But I think that the theoretical models tell us that this kind of boils down to the number of available beneficial mutations. So if there are lots of different options for improvement in one particular environment, then you're going to see more diversity of solutions in independent replicate populations. If there are very few number of available beneficial mutations, you have fewer options. There's going to be more overlap between replicate populations in terms of the genes that bear mutations. And we see some sort of relationship between the frequency of beneficial mutations and the degree of parallelism in my data. I will point out, though, that there's still a lot of variation. This isn't a super strong relationship. But for each of these environments, I'm only working with 3 replicate populations. So we have low sample size. So I think if we were to expand this experiment, we might have more power to see these kinds of patterns. So now I'm going to take a little broader view and ask, is this level of gene-level parallelism that I saw in my experiment, is this sort of typical for what we'd expect across a range of systems? There was this meta-analysis done a few years ago with Conte and colleagues that looked at gene reuse in natural populations. So looking at natural populations where similar phenotype adaptations occurred and looking at whether it was the same genes that underlay those adaptations. And they did see that, well, depending on the type of data that they looked at, there was about 30% or 50% gene reuse across populations. And we've looked at this sort of thing across microbial evolution experiments. So taking 16 different studies that were looking at the possibility of parallel evolution and taking all of those measures of the degree of parallel evolution, you can see there's a huge variety of different measures that we get. The mean is about 14%, but it ranges from 0 to 1, and there's a huge amount of variation. So the question is, again, what drives this variation? And we've done some meta-analysis to try and determine what kinds of factors are playing a role in the variation across these different studies. The first 2 that we found were playing a role are the number of replicates and the number of generations in those studies. This is maybe not so interesting. It sort of tells us that the more you sample, the more chance you have of seeing the same thing come up twice. The second 2 points here are perhaps more interesting. So there's a trend in these data that tells us that studies that use a larger population size tended to see a higher degree of parallel evolution. And then this goes along with our expectations. It makes sense because if you have a larger population size, it would expect to have a larger supply of mutations available. So whatever the most beneficial mutation happens to be, it's more likely to be there. You're more likely to have that fix in a particular population. You're more likely to have that fix in multiple replicate populations. If you have a smaller population size, things are going to be a little bit more stochastic. You may not have that best mutation within the population. And the final point here that we saw was that in those studies that worked with bacteria, we saw a higher degree of parallel evolution than in those studies that worked with fungi. So it was really just yeast. There were no other types of fungi. But for bacteria versus yeast, we saw this difference. And we're still thinking about what could be driving these differences. If anyone has any suggestions, I'd love to talk to you about that. Okay, so now I want to kind of get down to the fundamentals, the processes that are really at the core of parallel evolution and evolution in general, and then build this up and tell you about statistical approaches that we're taking to try and look at what are the important processes driving evolution by looking at sequence data from experimental evolution experiments. So the first thing that often comes up when you think about what drives parallel evolution is selection. So if you're thinking about a particular system, maybe there's one protein that's super important in combating a particular toxin in the environment, you might expect, if you have multiple independent populations evolving in that same sort of condition, you're going to tend to see mutations coming up in the gene that codes for that particular key protein, and that wouldn't be that surprising. What we're really saying, though, when we say selection is driving parallel evolution, is that the intensity of selection is variable across the genome. So this is just a cartoon that's depicting intensity of selection across the genome. And in this case, you see a lot of variation. You see some peaks. And at those peaks, those are the places where we might expect to see parallel evolution. Those are the places where selection is going to drive the mutations in replicate populations. On the other hand, if selection is not so variable across the genome, then there aren't really these hot spots where we might expect to see mutations coming up repeatedly in one population and another population. And there's another important process that we have to keep in mind, and that is mutation. So mutation rate can vary across the genome at different scales, and we know more and more about this over the last few years. But if mutation rate is not so variable compared to the variation that we see in the intensity of selection, then we can kind of ignore it and think about selection as the important process here. But the possibility is there that selection could be sort of a less variable, less important thing across the genome, and mutation rate could be highly variable across the genome. And under those circumstances, then it would be mutation that's playing that important role in driving which genes, which mutations come up repeatedly from one population to another. And it's this balance here that some of the methods that I'm going to tell you a bit about now tries to figure out, we try to figure out what's important, whether it's mutation or selection, and what's the balance between those two processes and driving the patterns that we see. So going back to the experimental evolution data, I'm taking the genome sequence data from the end of these types of experiments and then trying to ask two questions. The first is, can we predict which genes tend to be reused? So which genes come up over and over again in those replicate populations? And the second question is, can we quantify the relative effects of mutation and selection on the observed distribution of mutations? So meaning whether we see the same mutations coming up again and again in one particular gene or not. Okay, so here I'm just showing what sort of a picture of the data. You can imagine these are a bunch of different genomes from a bunch of replicate populations from an experiment. And the red marks are mutations that have arisen across that genome. If we pool all of those mutations, all of that data together, we can create a distribution. So this is all of the pooled information from, say, 40 populations. And you can see there's a lot of genes that bear 0 mutations. So along the x-axis, we have number of mutations per gene. There's a lot of genes that have no mutations, some that have 1 mutation, and then a few less that have 2 mutations, et cetera, all the way out to, for this particular data set, an example of a gene that comes up 21 times in these 40 populations here. So this is actually a real data set from Lang et al. 2013. So this is from Michael Desai's lab in Harvard, where they did an evolution experiment for about 1,000 generations and sequenced 40 populations for the yeast Saccharomyces cerevisiae. So using this kind of data with looking at a distribution of this type, we can ask the question, well, how did these mutations arise? Can we describe this process so that we can investigate how these mutations came about? And what we do is we describe this process as a Poisson process. It's a stochastic process, essentially just saying that mutations are rare events that occur across the genome. And we assume that they occur, and each gene has its own rate at which we are observing these mutations. And that's the lambda there. And the lambda, yeah, right. So, and the lambda is made up of two parts, the first being mu, which is the per gene mutation rate of that particular gene. And the second part is pi, which is the probability of a mutation in that particular gene being observed, basically. But you can just remember these as being mutation and selection. So we're interested in looking at these two parts, the first part being driven by mutation variation across the genome, and the second part being driven by selection variation across the genome. So I've taken two approaches, and for the first one, I'm using a Poisson regression. So using this base assumption here that I just told you about, we use a Poisson regression to try and identify genomic variables that can explain variation in pi, and so selection across the genome. So we've gone into the literature and looked through databases and got a bunch of variables such as gene level expression of genes, number of domains of the protein that gene codes for, some measure of codon bias, recombination rate, and a variety of other variables that we could pull out from the literature that we thought might be important in this process of selection. We've made the assumption that synonymous mutations are not undergoing selection, which we can talk about if you want to later, but this is a simplifying assumption that's probably true in most cases. And so that part, for synonymous mutations, that means that we've assumed that this mutation rate is constant except adjusted for by L, which is the length of the gene. So longer genes, we assume more mutations will arise in a longer gene than a shorter gene. For the non-synonymous mutations, we're then basically doing a Poisson regression, taking these variables that I talked about earlier and seeing, and using them to fit the model. So we go through and look for what the best model is. So what's the best combination of these variables that we've thrown into the model, or what's the combination that best explains the data. So for this particular data set, the Lang et al. Data set, we found a couple of variables that significantly explain the variation that we see. And those are gene length, number of domains, and recombination rate. I'm not going to go too much into the interpretation of why or why not these make sense. If you want to talk to me about it later, that's great. But I think the important thing is that you can find variables that significantly explain some of this variation. So we can start to take information like this and begin to predict which genes will tend to bear mutations. So which genes are going to come up, or going to tend to come up repeatedly if we have independent populations evolving. But unfortunately, it doesn't really explain all that much of the variation of the data. So you can see this is a pie chart that breaks down the variation in the data. There's a lot that's still unexplained. If we just assume a constant rate Poisson process without even thinking about those variables, those genomic variables, we can already explain 33% of the variation. And those regression variables actually only improve things by about 10%. So I'll tell you a bit about approach 2, which actually does a better job of explaining the total variation, but Here, instead of actually trying to understand the details of what drives the variation in these processes, we assume that there is variation in these processes, and we just want to try and characterize what that variation looks like. And the point of that is that we can then try to understand how much variation comes from mutation and how much variation comes from selection. So here I've said we want to find the distributions for mu and pi that best explain the data. So again, starting with this base equation that I talked about earlier. And we make some assumptions about the particular shape of those distributions. We assume that mu, so the mutation rate, is gamma distributed. So the mutation rate for each gene is just drawn from a gamma distribution. And the reason for the gamma distribution is simply because it's a flexible distribution. And this is a distribution that's been used previously for when thinking about heterogeneity and mutation rate. For the synonymous mutations, we assume that there's no selection going on again. For the non-synonymous mutations, we then try to fit distribution to this pi variable. And there's a number of different things that we've been thinking about, continuous distributions of various shapes. The simplest thing to start with is just looking at a discrete distribution. And the absolute most simple thing that we might want to start with is something that says there's, say, 3 categories of genes. Ones that bear deleterious mutations, ones that bear neutral mutations, and ones that bear beneficial mutations. And that's what I'm going to tell you a bit about now. So when we run that model on the same data set, this Lang et al. Data set, we can find the best fit parameters for these 2 distributions. So this probably means nothing, but put it up there anyways. We find the parameters for the gamma distribution. We can then characterize the discrete distribution of the selection coefficients, essentially. And we can see that this is a distribution that has some number of deleterious, a bunch of neutral, a lot of slightly beneficial mutations, and then a couple of really highly beneficial mutations. And then if we looked at the breakdown of the variation, again, in the same way as we did before, you can see there's still quite a lot of unexplained variation. But we've started to get a little bit better here, and we can actually break down this into how much we're explaining from the mutation process, so about 4%, and how much of this variation can be explained from the selection process, which is about 20%. Okay, so what have we learned there? So what I've told you about today is basically a selection experiment that I performed to look at the different, how much variation there was in parallel evolution across different selection environments. We found that there was quite a lot of variation, but it's not clear what it is about these selection environments that actually is driving these differences. We saw that the degree of parallel evolution can vary dramatically across organisms and environments. And while we can find genomic variables that help us to predict which genes bear mutation, it really doesn't give us a lot of predictive power. So there's still much more to be explained and understood about these processes. I reminded you that mutation and selection have the potential to play equal roles in driving patterns of evolution. And so it's important to think about both of these processes and how they vary across the genome. I quantified the balance of these 2 processes using statistical models based on this Poisson model that I showed you earlier. And for the particular data set that I talked about today, we saw that selection has about a 5 times greater impact compared to mutation. So from here, I think it's pretty clear that there's a lot still to be understood about what's driving the degree of parallel evolution and the variation that we see across organisms and environments. But we're starting to get a handle on this, and I think that using the statistical approaches that I've talked about today, we can go back and look at currently available data sets. So there's more and more experimental evolution experiments are being sequenced And it would be really interesting to try and take those and look at, apply these approaches and see how much variation there is from one organism to another and one experiment to another. I'd also like to try to set up my own experiments and directly test some of these potential factors that are driving the degree of parallel evolution. And so that's something that I'm going to be working on in the future. And these are some of the things I mentioned earlier that at the moment theoretical models are suggesting may be important. So they may be the first things to sort of focus on in these kinds of experiments. And finally, so I guess the ultimate goal is to try and use these theoretical models to try and predict evolution, which may be an impossible task, but at least we can try to understand when evolution may be more or less predictable. So under what conditions would we be able to predict whether a gene is going to bear mutations in a particular environment, and whether, and perhaps in other circumstances, we can say, well, it's pretty much going to be impossible to tell. And I've gone super quickly, but here I am at the acknowledgements, and so I'd like to thank my current postdoc supervisor, Thomas Bataillon, at Aarhus University, where I am right now, and a couple of people that have been working on these projects with me, Johanna, Francois, and Glo, and my PhD supervisor, Rhys Casson, at the University of Ottawa. If you have any data sets that you're interested in thinking about using these statistical approaches on, let me know. I'd be interested to talk to you. And with that, I can take questions for quite a long time, and then we can go for coffee. I can't see. Okay.
I was just wondering whether or not you think that gene flow might have a role to play in dictating parallel evolution in natural populations. I noticed you didn't mention it.
Yeah, so I've been thinking about populations where gene flow isn't happening. But of course, if you have, I guess maybe if you're thinking about gene flow of the same allele into the population, and then that, yeah, so under a situation like that, that's certainly going to be very important. And mutation's not playing a role there, but you want to think about other processes that are bringing those mutations into the population. So yeah, you could definitely start to try to incorporate that into these kinds of models as well.
Great, thanks very much.
Yeah.
I was wondering, hello. I was wondering about gene-specific variables that perhaps reflect function a little more, in whether you could model that with the specific selective pressure that was put on them, such as networks, metabolic networks, enzymes.
Yeah, that would be really cool. So I guess I don't have that information for some of the organisms that I'm working with. I mean, perhaps for yeast that's available, but I don't actually have that, or I wasn't working with it at least. It would be really interesting to have that information. I'm sure it could potentially be important in predicting. Yeah.
Thanks.
No problem.
Hi.
Hi.
So I saw a talk this morning about looking at Arabidopsis mutation accumulation lines, where looking at the fitness variance of these mutation accumulation lines, where the mean of the mutation accumulation lines was higher, variance was higher, but also the mean than the parental lines. And I was interested in that. It seems kind of unexpected. Similarly, your best fit model for the distribution of mutation effects, with this sort of bias towards slightly beneficial alleles, do you think that's biologically realistic? Because it sort of seems counter to what we expect. In your cartoon, you had neutral as the most prominent effect.
Yeah, that's true. Yeah, that's a good question. I guess I think that that's probably a drawback of these models. We're much less likely to be able to characterize the deleterious side of the distribution of fitness effects than the positive side. just because you're much less likely to ever encounter that kind of mutation in the data. That's certainly something to consider to try and adjust the models. Yeah, so I guess what I would say is that the distribution that I showed here is probably biased towards the beneficial side, and it's not realistic for the true distribution of effects. Yeah.